graduate.
Yeah, I'm now for like the last 2 days I haven't had it.
Well, college is a lot of dean and like associate deans and whatnot. I'm saying a lot of yeah, I
Program should have started now that we use it like said something. It doesn't make sense. We do like everything together. Like even down to like everything we've done together. And so I don't see why like she's having some issues with that.
The Iowa State. The Iowa State.
They are much better than they were before. It's like a jungle in there.
Yeah, I was thinking that conference lunch first. I think it's only like yes, I think it's just. I sounded exhausted.
Okay, we might just have to start without Dr. Okay, good afternoon, welcome, thanks for coming to Jodi's events, super exciting. So I've been Jodi's mentor for the past few years. She joined my lab after doing her rotations in fall 2020, and I was really excited that she started in my lab and had some computational expertise and really pushed forward a lot of projects. She did her bachelor's at Walla Walla University in bioengineering, and then she did her master's at Delaware State University. She studied gene expression in blueberry and looked at heat responses and transcriptome changes and things like that. So when she came, she already had like some really strong ideas and backgrounds on what she wanted to do, but I think over the last 6 years, we've seen her repertoire greatly expand and move beyond gene expression analyses to lots of different projects, which she's going to talk about today. She's been super active as a grad student, not just here at Iowa State in our Bioinformatics and Computational Biology grad program and in our department, Genetics, Development, Cell Biology, but also so she was recently awarded a National Corn Grower, which was really, I think, influential for her and gave her some experience with commodities and policymakers, which is a totally different side of things, but I think really, really awesome. She also was awarded an ISU Teaching Excellence Award, or a TEX. So for those grad students in the room, many of you know about the TEXs and the REXs, and these are really awesome awards. When it comes time for commencement, when I'll be sitting there crying, she'll be in her robe and she'll have her little extra cords on for TEX. We haven't heard about the REX yet. Hopefully she gets the REX too this semester. She's had conference travel support, and of course, I think the most notable was that she has been supported by Department of Defense as a SMART scholarship awardee. You know, Jodi was really, really proactive with funding discovery and applying for fellowships and trying to really get her own money in her first year or two with me. And the SMART was one of those things of like honorable mention and then go again. And then she got full funding, which is amazing. And then of course, this will lead into her next career stage where she works for Department of Defense, which I think is a great program. So Jodi has already published a couple papers on her research. She's had other awards too. There's too many, there's really like 2 pages here. She's already published a couple papers, which she will talk about today. And one of them also I think is unique because it highlights her work in the community for being like really influential on pushing forward policies and ideas related to being a good data steward, and working with the Ag Bio Data Group, which is something that she totally just did on her own, had nothing to do with me. She's very independent. What else do I want to say? Okay, she's also a fantastic communicator, which you'll see today, for those of you that haven't heard her talk yet. But she has been invited speaker at fantastic conferences like Plant Animal Genomes, the GRC Plant Proteolysis in Italy, and many of the North American Plant Phenotyping Network conferences, where she has also been keynote and invited speaker. So she's going to tell us about her research today. I'm going to turn off the lights just because I think it will help with the slide. And biological network. Introduce myself a little bit. I'm from Maryland, also known as the DC, Maryland, Virginia area, or DMV. So fun fact, I know everywhere else DMV means the place that you to get your license and stuff. But in Maryland, where I'm from, we call it the MVA, the Motor Vehicle Administration. So that was like a culture shock, and I'm like, I'm from the DMV, and they're like, you're from the driving test? No. Just some random facts about me. I am a gamer. I like roguelikes and shooters. I'm in a Marvel Rivals team. I enjoy anime, and I play like D&D at least once a week. Over my time here, I've been very busy, and I have a lot of different projects, but they break into 2 categories, mainly being automated phenotyping and biological networks. So we're going to talk about both of those together, but first we're going to kind of start at the beginning of all things. So first of all, why maize? Maize is a really pivotal crop, and it supports a large part of the developing world's calories and also a lot of animal calories. It's also a really big part of our economy here in the US. So it is an extremely important commodity. And one thing that is really pivotal in understanding the way plants grow is understanding phenotypes or their physical manifestations. And that's impacted by the environment and also underlying genetics. Now, there are many ways to assess phenotypes. There's a phenotyping group for every part of a plant, from shoots to tassels to roots, all of that jazz. But in the Kelly Lab specifically, we study roots. Now, that's a little difficult because roots are soil bound, they're hidden, and they're very difficult to get to. So part of what's very interesting about this work is kind of looking at like a hidden world underneath the soil. So throughout my time here, I use a lot of omics, which omics is a very fancy way of saying a mode of measuring biological data. And a lot of my analysis was multi-omics, which is analyzing 2 different types of biological data simultaneously to look at a particular problem. So during my time here, I used many different types of omics, including transcriptomics, genomics, proteomics, phenomics, and interactomics. Now, there are several different types of analysis methods, and they broadly depend on a lot of factors, like the type of omics that you're using, how closely related the data is, if there are treatments involved, and how noisy your data is. And there's different transformations that can be done, different visualizations or clustering methods that can help in analyzing that data.
largely what has been really cool in recent years has been the advent of machine learning tools that has made it a lot easier to identify sort of pockets of information that are sort of correlated together that we can further analyze. And I'll talk more about that throughout my chapters. I wouldn't have been able to accomplish any of this work without all of the great scientists and researchers who came before me who made tools that made analyzing this easier. A big shout out to the people at Jodi B. They're on campus. I've met with them. They are amazing people, and they have been really supportive of my research. And also thank you to Uniprot. I think I crashed them one time, so thank you so much for your patience. So for my particular work here, we're going to talk about like novel tools. We're first going to be talking about the imaging parts and the phenomics, and then we're going to talk about some predictive network development as well. So methods in plant phenomics, this is a submitted chapter under review currently. So roll towel assays, what are they? They are a method for scientists to look at early development stages in plants, particularly in the kill lab. We use them for maize. This is like a very staple assay that happens in the Callwood lab, like go through hundreds of these in a week, maybe. In order to extract the phenotypes, somebody either has to sit there with a ruler and write down like what the measurements are for whichever type of fruit that they're trying to measure, or take an image and someone sits in image J and then measures the roots, right? So imaging has definitely helped move that process along, but we can do better. So introducing automation with something called Root Painter. So Root Painter is a segmenter, and it is built on the UNet architecture, and it can be trained to separate desired structures from the rest of the image. In our case, we're interested in the roots, so separate the roots from all of the rest of the image. And so Root Painter was able to be trained to do that. And then using a Rhizovision Explorer, it's a very popular, well-known root phenotyping extraction tool. And phenotypes from these roll towel images, we're able to do that and analyze it. It took about 2 months to train Root Painter fully, and this was 2 months by an undergrad that I trained to work on Root Painter. It's very user-friendly, so that was great. But it took him 2 months. He had no prior computational training before he did this. And we are currently using it in lab, and we can run about 40 images in 40 minutes. So much faster than having an undergrad measure these by hand or having a scientist in the lab just like rolling out towels and jotting things down in a notebook. And here we can see more closely the progression of training Root Painter. So at the far image, what's in blue is kind of what Root Painter is selecting as significant. The green brush is background, and the red is whatever known structure that we want, in our case, roots, right? So over time, we can see that Root Painter is getting better and better at selecting just the root structures. Here, it's having a little trouble with some of the more fine lateral roots, but it gets corrected here to an almost perfect sort of segmentation. And we've made this process, it's very fast, but there have been some improvements in technology. So in collaboration with Clarice, who is like the bench grad student in our lab, we worked on optimizing the ring light sort of light concentration for the roll towels to make the assay capture very uniform. Also, you can run an image like this. This segmented image is not like perfect, but you can run it in Rhizovision if you wish. you would just need to make sure that your diameter thresholding is exact so that it only captures the root structures. But Microsoft has been trying to put AI in everything, but this is actually kind of good. They put AI, they have an AI photo eraser in their photo app. And so we're able to select a large swath of the image in order to clean this up fairly quickly. So if you're willing to spend some extra time sort of like pruning those images before putting them in RizoVision, you won't have to do the diameter thresholding. So it's like a trade-off. You either have to do diameter thresholding or you pre-process the images. Okay. And that pipeline has gone through many iterations. Currently, there are 2 instances of it. One works for the roll towel images with the seedling roots, and the other one works for crown roots. And today we're going to talk about the crown root application here. And this paper is published in Plant Phenomics, but what we did was train a root painter RizoVision pipeline instance on crown roots, extracted those phenotypes, and used transcriptomic data and analyzed it in conjunction, sort of like a side-by-side analysis, to see if there were any like pockets of expression for things that were significant for root development. And this project started with a previous grad student in our lab, Craig Cowling, well, Dr. Cowling now, who observed that different genotypes in maize, when they were grown, like their root structures are different even though they're closely related. Of course, they would react differently between the mock and the auxin treatments, but even between just different genotypes in the mock, we can see different root development at like the roll towel stage. But we're also seeing that as well in the field. So the question is there a correlation between what we're seeing in the early stages to what we're seeing in the field stages? Can we sort of predict what our root structures may look like? And here's an application of the pipeline trained on the crown root images and segmented RizoVision. And with the data extracted from RizoVision, it extracts around 27 phenotypes. We compare them to B73, which is our benchmark. And the stars indicate statistically significant phenotypes for B73. So there are, as compared to B73 in those phenotypes. So there are several traits that are significant across many different genotypes, but that's still not enough. We want to understand why that they're different. And so we look at some of the transcriptomic expression. So here we have the top 100 transcripts compared to B73, and you can see that there are several pockets of very high expression in many different genotypes, and some also pockets of low expression. So we'd like to investigate those more, and in order to do that, we did a WGCNA, or a weighted gene co-expression network analysis. And this is one of the pockets that we, one of the clusters that we looked at. And this particular cluster is related to the genotype W153R. And here, what's interesting is that we were able to get 2 go-aheads for beta-134-glucan, which is an enzyme that is necessary to facilitate cell wall loosening, and that enables root growth. So we can see in a genotype that had some heavy root development expression, we're seeing an enrichment in a root development in an enzyme that is correlated with root development, and that is very interesting. Hope to look into that more one day. Maybe not me, but someday. So back to our central question, is there a correlation between sort of the roll towel images like the seedling development and the field development? And the answer is kind of, which is consistent with the literature. Some genotypes show a very strong correlation and some don't. So that's kind of what we got here. Some very strong correlation, others not so much. But with all of those, with our new pipelines, we can extract a bunch of phenotypes. We can do a lot of cool things with those phenotypes. And one thing that I really wanted to do was transcriptome-wide association study, or TWAS. So broadly speaking, TWAS is a method that correlates transcriptome abundance with a phenotype in order to narrow down which transcripts are pivotal to that phenotypic expression. Different packages have different methods of doing this. For this particular application, I use the Bravo package, which is based on Bayesian trees, and that was developed here at ISU in our lovely stats department. So I used the pipeline that for the roll towel images, and the Kelly lab did a derivative of the WEDIV study looking at genotypes that were strongly correlated with root development. And so extracted a bunch of those roll towel images. And this is a sample of the types of phenotypes that we find. There are a lot of root tips and branch points that they vary by genotype. So it goes high, medium, low. So here we can see a bunch of branch tips and root tips and branch points. It might be a little hard to see in the back, but there are a lot of lateral roots offshooting here. So this would be considered a very high expression of root tips and branch points. Here it's kind of in the middle, where some of them have quite a bit of root tips and branch points and others not so much. And here we see very few root tips and branch points with not as much lateral root development. So we did this for around 200 genotypes. I also did transcriptomic analysis for those 200 genotypes, and we were able to cluster like the top 100 transcripts and found some really cool clusters. But I think this would make a really cool glass mosaic. So if anyone wants inspiration, here you go. Please take a picture and send it to me. But with that data, we're able to put those extracted phenotypic traits and our transcriptome data into Bravo and perform a TWAS and get some hits per the different traits, right? So we got several hits, which are these transcripts that seem to be pivotal for these particular traits. And one that was of particular interest to us is Eskimo-1. Eskimo-1 has been shown, like in Arabidopsis, to be involved in water economy, drought, stress, and salt tolerance. Those are also facets that are strongly correlated with root development. So it would be very interesting to study it in maize as well. And so we did a CRISPR on Eskimo-1 in maize. And shout out to Clarice, who tried her hardest to keep these plants alive. But it seems that the CRISPR was seedling lethal for Eskimo-1. So that could be due to like CRISPR making them allele or something. But there are many different methods of like testing this particular trait, and we'd love to test it again, maybe in the future sometime. But yes, it was seedling lethal. So in short, Eskimo-1, of course, the phenotype of seedling lethal, it does warrant some additional study. But the Bravo package did identify many other transcripts that could be significant, and so it's worth investigating some of those other ones as well, and the traits associated with them. So now we're going to like switch gears a bit. That was my phenotyping work, and now we're going to talk about the networking stuff. And we're also switching organisms. We're out of maize, we're into Arabidopsis. So, Arabidopsis. Arabidopsis is a model plant organism, and we're gonna also talk about auxin, which is essential hormone for growth and development in plants. So one of the really cool things about auxin is that across different cell types and tissue types, for instance, in a root, it can create like a gradient, almost think of like an ombre color concentration of auxin levels, and that triggers different sort of stages of development in the different cell tissues. So the question is, does this impact how like kinases are, like what kinases are present in these different cell types and sort of their behavior? So what is a kinase? And so a kinase is a type of enzyme, and it transfers a phosphate group from high energy donor to a specific substrate in its active loop, that little loop-de-loop right there. So we're looking at kinases and their corresponding substrates in the different tissue types. So we're looking at the endodermis and the epidermis, and shout out to Dr. Buss for taking these great pictures. So what was the approach to do this? The approach was using proteomic data generated by the Wally lab to activate, look for activated loop kinases, and using a published correlation matrix that used Pearson and Spearman correlation to identify likely interactions between kinases and substrates, visualizing those networks, and then doing a Go enrichment analysis per tissue type. So this is the network of predicted kinases. We're here we're looking at, like, the blue is the epidermis versus endodermis, and the green is the epidermis, the pink is the endo, and the orange is epi plus endo. So the orange one are ones that are shared between all of the tissue types. So we can see that there's only a handful of sort of kinase nodes that are shared between all of them. And here are some different subnetworks that's a little bit easier to see. The blue are the kinases, and the vermilion or pink are the substrates. And so we can see that in the different tissue types, there is, some of the networks are very small and testable, and some of them are very expansive. But we can see a trend of there being very few kinases and many substrates that interact. So overall, auxin has varying impacts on different root cell types, and there are different kinases and substrates interact, and there's like many different groups, and there needs to be more studied on this for testable interactions in order to like get these networks down. And also the Wally lab has more updates. So I will now talk about a project that took up a lot of my time, but it's near and dear to my heart. It is Puppet. Shout out to Applebee's, because that's where Clarissa and I came up with this name. So Puppet is a predictive pipeline that looks at ubiquitination, and it predicts if an E3 and a substrate interact, and if that interaction results in ubiquitination. So what is ubiquitination? It is a form of post-translational modification, which changes the behavior, or quality, or location of a protein. And this process happens in multiple steps. The step that we're most interested in is this step here between the E3 and the substrate. There are around 1,300 identified E3s in Arabidopsis. And Song from the Wally lab created a catalog that identified over 17,000 ubiquitinative peptides in Arabidopsis. So it's a catalog of substrates, essentially, that was identified. And so now the outstanding question is, we have around 1,300 E3s and around 6,000 substrates. And so which E3 goes with what substrate? And that's where Puppet comes in. So there was a published work that used a Bayesian network to identify E3s and their corresponding substrates in humans. And so I took that concept and I added onto it by adding a binary layer, co-expression layer, as well as two protein prediction machine learning tools, which is HDoc and ESM-Fold. Both of them are sequence-based tools in order to predict, well, with high confidence, if two things are interacting. And so this is Puppet, which is the predictive ubiquitination protein pipeline for E3 substrate interaction testing. And it is a function in the Jutsu package from the Hidden Maze Village. So if we just did the Bayesian work, we would end up with a network like this. Still smaller than with 1,300 times 6,000, right? Still much smaller than that, but this is still not manageable for testing. And in comes Puppet. So we did a lot of different networks, but this is just a subset network. This is for specifically the protein group that is the FBox network. With our Bayesian network, we have 52 nodes and 48 edges, which comprises of 7 kinases and 45 substrates. But with Puppet, we get a much more manageable network that has 13 nodes and 11 edges with 2 kinases and 11 substrates. And this is a sample of kind of what that analysis looks like. This is between FBox and Gout10. Gout10 is very interesting for us in the Keller lab because it is an enzyme that helps build cell wall components, and cell walls are very important for root development and root elongation, root growth. So this was very exciting to find, and this unnamed FBox here, and this is some results. This is a contact map that was generated from ESM-Fold. This this interaction met our requirements for HDoc. But here is the contact map, and we can see that more like towards like the middle of this particular protein, we're seeing a lot of contact. And due to computational limitations, the proteins had to be split in order to run this, because this protein is very long, this unknown FBox protein. So something to think about in the future would be how to split proteins for computational purposes without disrupting domains, which is something I was thinking about. So that is, here we have these contact maps, very great. And this is an example of kind of what you would get from HDoc. So the blue is the primary structure, and the green is the secondary structure, and the alpha helixes are the vermilions, and I really like this protein structure. The box, the Golgi marker, we can see a merge, which means these two things are interacting in some way. What the result of that interaction is, we're not quite sure yet, but they are interacting, which is cool. And so in summary, Puppet is an effective pipeline for narrowing down those interactions paired to E3 substrates. There still needs to be more work done in testing those interactions to see how accurate Puppet is, but still with a giant leap forward that we're able to get some manageable networks. And finally, I'd like to talk about something near and dear to my heart, volunteer work with the ag bio data. So I had the privilege of being part of a phenotyping working group where we talked about how to properly store, manage, and access phenotyping data in a way that would benefit the scientific community. And so we wrote a white paper for fair phenomic databases, advocating for resources and support for building a phenotyping database and what that would look like. And that is published in Science and Public Policy. And finally, I'd like to thank my lab mates. I'd like to thank Dr. Kelly. Without her, none of this would be possible. I'd like to thank Danny for doing a lot of the lab work, and Clarice for keeping the plants alive, and Lauren for hanging out, and Ella, who's not here, but she is like Clarice's right hand, been extremely helpful. And Owen is my undergrad that I've been training, and he's only a sophomore, but he's very bright, and he's been doing a lot of really good work, and I'm very proud of him. Also Amanda, who has been helping Gammy quite a bit. Also our collaborators, I think Dr. Wally and Shuka and Song for all of their support. Bus for the beautiful pictures, and my sponsors from DOD, Emily and Julie. I'd like to thank my committee members as well as former lab members. A special shout out to Dr. Cowling for all of his great work, and Jasper and Jacob and Luke, who I mentored and who helped a lot. And for Jackson for shoveling up all of the roots because I was not going to do it. And my funding sources as well. So thank you for your time and attention. Are there any questions?
Okay, so we'll do questions from the audience, and then committee will save their questions for a little bit. Dr. Greenberg.
Thank you, Jodi. Great done. Congratulations. This far. Fantastic. I have a question about, I think it was chapter 3 of your piece. When you do the associations between transcriptomics and phenomics, so I assume that initially you split, you kind of get a set of phenotypes, say, I don't know, I'm just throwing a number, let's say 10 phenotypes, okay, or 20 different phenotypes, and then associate your transcripts with that.
Mm-hmm.
My question is as follows. I know very little of phenomics, so this might be totally out there. How do you, how fine-grained or coarse-grained do you differentiate between different phenotypes? Like, is there like a set rule of these, what I see here breaks up into 20 phenotypes, or into 30 phenotypes, or 10 phenotypes? Is there an objective way of assessing that? And if there isn't, as I suspect, have you tried kind of, different ablations or removing or trying different phenotypes and seeing how robust your analysis is based on the number of phenotypes actually that might be there, might be 10, might be 20, depending on how you differentiate that? Does the question make any sense?
I think it does. So what I'm hearing is, how do we define phenotypes, and have we tried different categorizations of phenotypes to optimize our analysis? Is that right?
Yeah, so have you just used different categories of phenotypes and seen how different that transcriptomics that explain them are? Yeah.
Yeah, that's super interesting. The answer is, okay, so the first one is root phenotypes rhizovision for our analysis. Some of the phenotypes are more reliable than others because we're doing 2D images, so some metrics like volume and things like that are a little less reliable because it's a 2D image. But if we were to use the same transcriptomic or data, well, no, because it's on roots.
You could go to your slide